This is a lecture on the cell cycle and mitosis for Bio 101 for September 28th, 2020. And here we can see the overall process of mitosis taking place and one cell being made into two. And we're going to discuss the stages of the cell cycle as well as how the cell cycle is regulated and the stages and hallmarks of mitosis, or the actual cell division, in today's lecture. So first, why should we care about the cell cycle? So cells in your body need to divide or make more cells for several reasons. The first one is, as a multicellular organism, we need the cell cycle in order to grow. We need more cells for growth to occur. We also need to be able to replace any old or damaged cells in our bodies and fight infections. And so the immune system is constantly making more cells that can help us fight infections and pathogens. And it's important that this process of cell division, or making one cell into two via the cell cycle and mitosis, is done correctly, or the cells can divide either abnormally or incorrectly, or uncontrollably, and lead to cancer. So what is the cell cycle? It's basically just a sequence of events that a cell needs to pass through between being one cell and becoming two. So between one cell division and the next. And so it's how one cell becomes two. And it's split into two major stages. The first being interphase, which you can see here in purpley blue, and the second being mitosis, or the M phase, which is seen in red here. And for this part of the lecture, we're going to focus on the stages of interphase, and then we're going to have a separate lecture just on M phase itself. And so as you would notice, the cell spends most of its time in interphase, especially compared to M phase. And so we're going to talk a little bit about the stages within interphase and what happens in each of them in the next slide. Right, and so as I mentioned, cells spend most of their time in interphase, right? And some cells actually never even complete the full cell cycle. And they'll enter into this non-dividing state known as G0. One type of cell that does this is neurons. So some neurons are non-dividing and will not complete the cell cycle. And they permanently exist in G0. And G0 cells are perfectly normal physiologically. They still carry out their functions. They just don't divide, right? And so they don't go through the rest of these phases of the cell cycle. The interphase stage of the cell cycle is divided further into three separate phases called GAP1, or G1, S, or synthesis, and GAP2, or G2 phase. So we've got G1, S, and G2. And so in the first phase of growth, or GAP1 phase, or G1, cells make macromolecules. And we've already discussed macromolecules, proteins, carbohydrates, and lipids as the building blocks of cells. And you can imagine that if one cell needs to be made into two through the cell cycle, we're going to need the pieces to do that. Another cell is going to require more proteins, more lipids, more carbohydrates to become two cells, and so those need to be synthesized and made, and that happens in the G1 phase of the cell cycle. And then moving on through interphase past this checkpoint, the cell will enter into S phase, or a synthesis phase, and this is where the DNA doubles or replicates. And so once again, because we're going to take one cell and make it into two, we need twice the amount of DNA as well. So we've already made twice the amount of macromolecules in G1. We're going to double the DNA to make enough for two cells in S phase, or synthesis phase. And then once the replication is complete, so the cells can enter into G2. And in G2, or GAP2 phase of this cell cycle, the DNA that was just replicated in S phase gets checked, and repairs are made if they need to be. The cell doubles any organelles that it's going to need to divide from one cell into two. So right, we've got to double organelles as well. It additionally makes extra membrane that will be needed to make one cell into two, and then it can ultimately enter into mitosis, or the M phase. And so what's important to remember about the cell cycle is that each step of the cell cycle is checked and double checked and triple checked and is extremely highly regulated, right? So you'll notice here in this diagram there are several checkpoints that I've highlighted in red. There's a checkpoint between G1 and S of interphase. There's another checkpoint between G2 and S, as well as a checkpoint in the middle of mitosis. And this regulation of the cell cycle is important because you need to be able to make sure that if you're going to take one cell and divide it into two, there's enough components there, right? You've made enough proteins, you've made enough enzymes, enough organelles, you've correctly replicated the DNA, so that when you divide that one cell into two, each of those two new cells is healthy and has everything it needs to survive. And if that's not the case, the cell cycle will stop at one of these checkpoints, right? And so at the first checkpoint, sometimes this is known as sort of an external check, right? And there are a couple questions that the cell needs to answer in order to move from the G1 phase to the S phase. And the first one is really, does the cell have enough food? Does the environment supportive for making a new cell? Is there damage to the DNA? Because if there is, it might not be a good idea for that cell to divide and pass DNA damage on to a new daughter cell. And additionally, is there a growth factor present? And what a growth factor does is it basically promotes entry into the cell cycle. And growth factors are normally released by other cells to signal your cell to enter into the cell cycle because everything is good, there's enough food, the environment is favorable. And so if the environment is good, if there's a growth factor present, and if your DNA is not damaged, cell passes this G1 checkpoint and moves from G1 into the S phase, where DNA replication can take place. And then after DNA replication, there's sort of a checkpoint in the middle of G2 that makes sure your DNA has been fully replicated. If there's any errors there, that they are fixed. And if there's enough organelles to make two new cells. This is sometimes known as more of an internal check, right? So in the G1S checkpoint, we were checking the environment to make sure that it was favorable for growth. And here in G2S, we already know the environment's good. But what we want to make sure is that we have enough DNA and enough molecules as well as organelles to make two new cells. And that if there are any errors in the DNA, that we take the time to stop at this checkpoint and fix them before moving on into mitosis. And so what happens to a cell if it doesn't pass the checkpoint? If it can't say that the DNA is not damaged or if it's not sufficiently replicated? What happens to the cell? And so some cells will just stop at that stage of the cell cycle and never divide. They'll become what's known as arrested or paused in that stage indefinitely. However, some cells will undergo a process called apoptosis, which is programmed cell death, or it's like a cellular self-destruct button, right? And rather than continue dividing and creating cells that might not have enough molecules, organelles, or might have damaged DNA in them, the cell will choose to not divide, but rather to die, so as to save any future cells being created that don't have the proper components or that have damaged DNA. And so throughout life, there's this balance of cell division, or the cell cycle, and apoptosis, or cell death. And so you can imagine that in order to grow, we need cell division to be increasing, right? But at the same time, we need to be able to destroy damaged cells so that they don't infect the rest of the body. And we also need to destroy cells as we develop over time. And so some examples of this are sort of in organ shaping and tissue development. Apoptosis helps with the removal of tissues that are unnecessary, like the tail of a tadpole, as that tadpole develops into a frog, as well as to sculpt organs. And in particular, I like to think of this example as the perfect reason to understand why apoptosis exists, right? And so as we develop as humans, we actually have webs of cells in between all of our fingers and toes. And those cells are not necessary. In fact, they would actually be a hindrance to our proper kind of hand formation or hand function. And so those cells need to be removed safely to basically allow us to have free digits and no longer have webbed hands and feet. And so these unneeded cells undergo apoptosis in order to kind of sculpt our hands and allow us to have digits that we can use, right? And so any abnormal or damaged cells, as well as cells that are just no longer necessary, will undergo apoptosis. And there has to be this balance of making cells via cell division and killing cells that we don't need via apoptosis. And so I just want to take a couple seconds and review stuff we've gone over in this lecture. And so take a minute, pause the video, and figure out which one of these is correct about interphase. All right, so D is the correct answer. Interphase does take up most of the cell cycle. It includes the 3 stages, G1, S, and G2, and it ultimately results in an increase in cell size because you're making more molecules, you're making more organelles, and so the cell needs to grow and almost double in size so that it can split into 2 new cells. Take another second, pause the video, and select the correct sequence of stages in the cell cycle. And the correct sequence is D, G1 or GAP1, S or synthesis, followed by G2, GAP2, and then M or mitosis. And finally, which of these is true about apoptosis? Take a second, pause the video, and pick your answer. The correct answer is D again, right? And so apoptosis is a form of programmed cell death. It does free fingers and toes of human embryos from that web structure by eliminating the cells in between. However, it's not a process that increases the number of cells. Apoptosis is cell death, and so it decreases the number of cells. And so in the next part of the lecture, we're going to discuss M phase or mitosis in more detail.